Howdy, my name is Adamari de la Cerda, and my partner Mario Flores Castro and I will be discussing our fifth experimental lab report on the IMViC test that includes the indole, methyl red, Voges Proskauer, and citrate tests. To begin, IMViC refers to a set of four biochemical tests that help differentiate bacteria, especially bacteria from the Enterobacteriaceae family. The indole test ultimately tests for a bacteria's ability to convert tryptophan to indole with the presence of a sulfide indole motility agar. Methyl red tests for the ability to perform a mixed acid fermentation. The Voges Proskauer tests for the production of acetylethylcarbinol. Lastly, the citrate experiment tests for a bacterium's ability to use citrate as a sole carbon source. In order to initiate the indole test, the bacteria needed are E. coli, E. cloaceae, and the negative unknown 201. To begin the indole test, first obtain a sterilized test tube with 4 ml of tryptophan broth. Then aseptically inoculate the respective bacteria. Lastly, the samples will be incubated and the results recorded. The indole test for E. cloaceae displayed no color change or movement in addition to a clear broth. The same results were witnessed with unknown sample 201 except with the presence of some turbidity. Ultimately, E. cloaceae and the unknown sample 201 were interpreted to be negative results. On the other hand, E. coli displayed a red color change towards the surface of the tube, in addition to some turbidity. Therefore, E. coli is interpreted to be a positive sample for the indole test in converting tryptophan to indole. In order to complete the methyl red and Voges Proskauer tests, the bacteria needed are E. coli, E. cloaceae, and the negative unknown sample 201. To initiate the test, first aseptically inoculate the respective broths with the selected bacteria. Lastly, the samples will be incubated and the results recorded. The methyl red test for the unknown 201 sample displayed a red color change in addition to the presence of turbidity. On the other hand, E. coli and E. cloaceae both displayed a red color change with transparent broths. Ultimately, all three samples were interpreted to be positive for the methyl red test in producing mixed acid fermentation.
The Bogues Proskauer test for E. cloacae and E. coli showed negative results, as the broth remained clear, yellow color. This means that these bacteria do not produce acetoin after glucose fermentation. Unknown 201 also showed a negative result, although there were bubbles in the surface, which we concluded were just bacteria that precipitated. It is important to note that this sample was supposed to be positive, which means that it does form acetoin, and it should have been a red color. For the citrate test, we used E. coli, E. cloacae, and our negative gram sample unknown 201. The procedures for this test were just to inoculate the citrate agar by striking the slant with a loop. Then we left it to incubate for a few days and then we record the results. For the citrate test, unknown 201 showed a positive result. This is seen in the color change from a green to blue. There was also growth present on the slant. This means that unknown 201 is able to metabolize citrate. E. coli and E. cloacae were negative results, as there was no color change and there was no growth. In conclusion, the IMViC tests are four important lab tests used to differentiate bacteria, mainly Enterobacter and gram-negative bacteria, by testing on their metabolic abilities. The four tests are indole test, methyl red, box Proskauer, and citrate test. After running these tests, this is what we concluded about unknown sample 201. For the indole test, we saw a negative result, which means it does not metabolize tryptophan. For methyl red, we saw a positive result, which means that unknown 201 produces acid as a result of glucose metabolism. For both Proskauer, we saw a negative result, although it should have been a positive result. For the citrate, we saw a positive result, which means 1 is able to metabolize citrate. Thank you.